Biochemistry 401, Lecture 4. Today we're going to continue our discussion of basic concepts. We're going to revisit basic thermodynamics. We'll start with an introduction to open and closed systems, and then we'll continue our discussion of the carbonic acid bicarbonate blood buffering system. We'll then talk about the first and second laws of thermodynamics, and we'll discuss Gibbs free energy. The equation for this is listed below. So let's get started. So let's start with a basic introduction to thermodynamics. What is thermodynamics? Thermodynamics is a study of the relatedness between heat and power. This comes from the Greek therme for heat and dynamis for power. Thermodynamics examines the physical nature of energy and force. And some of the key concepts in thermodynamics we've already touched on in the last lecture. These include the system, the surroundings, the universe, entropy, and enthalpy. We're going to talk more about these in more detail in this lecture. And so let's go over the concept of system and surroundings one more time. A system can be anything that you are studying. It's the place where the reaction is actually taking place. It can be a beaker or a subcellular compartment. It can be a thermal vent or even the Earth itself. And the surroundings is everything in the universe except for the system. And so therefore, the universe is a system and its surroundings. So the system is the area of interest where the reaction is taking place. The surroundings is everything else in existence, excluding the area of interest. And the universe is the system and surroundings. So now let's discuss some characteristics of systems. There are many types of systems, but we're going to consider two of them. In this diagram, you'll see an open beaker and a closed Erlenmeyer flask. In an open system, heat energy can be exchanged between the system and its surroundings, and matter can also be exchanged between the system and its surroundings. Things can be put into the system and things can leave the system. On the other hand, as with this closed Erlenmeyer flask, which represents a closed system, heat can be exchanged between the system and its surroundings, but matter can't get into the system, and matter can't leave the system. And that's the difference between an open and closed system. The difference is whether matter can leave or enter. You will recall that the carbonic acid bicarbonate buffering system has a pKa of 6.1, and this is the buffering system that we use in our blood. So why does this work so well? Well, one of the reasons is because, like other living things, our bodies represent open systems. Let's look at this a little more closely. Now, metabolically active tissues create a large amount of carbon dioxide. When carbon fuels are broken down, carbon dioxide is one of the waste products of this catabolism. And you say this by saying high partial pressure of carbon dioxide, because carbon dioxide is a gas. Anyway, metabolically active tissues produce a high amount of carbon dioxide, and so they have a high partial pressure of carbon dioxide. So when there's a high concentration of carbon dioxide, this tends to move the reactions that you see below towards the right. Carbon dioxide enters red blood cells, and there it combines with water to form carbonic acid. Now, because the pKa of carbonic acid is 6.1, and the pH of the red blood cells is somewhere between 7.35 and 7.45, this causes the carbonic acid to become deprotonated to bicarbonate and a proton. This bicarbonate leaves the red blood cells and travels in the bloodstream to the lungs, and in the lungs the partial pressure of carbon dioxide is low. That's because we continuously breathe off carbon dioxide. And so because there's a low partial pressure of carbon dioxide, these reactions that you see down here at the bottom tend to go in the reverse direction. The equilibrium is skewed to the left. And so bicarbonate takes up a proton to become carbonic acid, and then this is broken down to become carbon dioxide and water, and these are both breathed off as carbon dioxide gas and water vapor. And so to go over this again, in the tissues, carbon dioxide and water that are produced during metabolism leave the tissues and enter the bloodstream. The carbon dioxide and water go into red blood cells, and there are enzymes in the blood cells that convert carbon dioxide and water into carbonic acid. Carbonic acid is rapidly deprotonated to become bicarbonate and a proton. The bicarbonate then leaves the red blood cells, but it still stays in the bloodstream, and it travels to the lungs. And when it's in the lungs, it becomes protonated to carbonic acid, and then carbonic acid is broken down to release carbon dioxide and water, and these products are released as carbon dioxide gas and water vapor as we breathe. So by increasing and decreasing products and reactants, we can skew reactions to the left or to the right based on Le Chatelier's principle. I'm sure you remember that from chemistry. Okay, so let's look at this a little more closely. When there's high partial pressure of carbon dioxide, high reactants, these reactions will tend to go in the rightward direction. Carbon dioxide and water making carbonic acid, and then that forming bicarbonate and a proton. When there are low concentrations of products, in this case bicarbonate, that also tends to drive the reactions to the right. On the other hand, when there is low partial pressure of carbon dioxide, when there are low reactants, this low partial pressure is going to drive the reactions to the left, and we'll have carbon dioxide produced. And when there are high concentrations of products, this tends to drive the reactions to the left. There's a characteristic ratio of products to reactants that you see at equilibrium. When reactions are not at equilibrium, there's a higher concentration of products or a higher concentration of reactants. So by increasing the amount of products or by increasing the amount of reactants, you can skew these reactions to the right or to the left. This is one trick that we use quite a bit. Living things tend to keep reactions skewed by changing the concentrations of products to reactants in order to get reactions to go one way or the other as we need them to do at that particular time. It is very important to maintain blood pH within a specific range between 7.35 and 7.45, and so blood pH is monitored by chemoreceptors that are found in blood vessels like the carotid artery and in organs like the brain and in the kidneys. These signals are sent to the lungs and to the kidneys to help modulate blood pH by modifying the exhalation of carbon dioxide and by modulating the elimination of bicarbonate in the urine. And so we said that the normal pH range in the blood is between 7.35 and 7.45. Now above this pH value, the blood is said to be alkaline, and this is a condition called alkalosis. Below this pH, the blood is acidic, and this condition is called acidosis. The body has two major ways of regulating pH. One is through regulating the rate of breathing. When breathing increases, more carbon dioxide is eliminated, and so the pH increases. Likewise, when breathing rate decreases, less carbon dioxide is eliminated, and the pH decreases. On the other hand, a slower way to regulate pH is through the regulation of bicarbonate elimination, and this happens through the regulation of the kidneys. When bicarbonate elimination in the urine decreases, the pH of the blood will increase. And when bicarbonate elimination in the urine increases, the pH will decrease, because again, bicarbonate represents the base that's found in the blood. So now let's shift gears a little bit and talk about the laws of thermodynamics. The first law says this: the total energy of a system and its surroundings is constant. This was proposed by Julius Robert von Meyer in 1841 and also by James Prescott Joule at around the same time. That says that energy is neither created nor destroyed. And another way to say this is, considering that E means energy, the energy of the universe is equal to the energy of the surroundings plus the energy of the system. And the energy of the universe is constant. This is the first law of thermodynamics. The second law of thermodynamics says that the entropy of the universe always increases. The entropy, as we said, is a measure of disorder or freedom or randomness. The entropy of a system plus its surroundings always increases. Large ordered structures will give rise to smaller structures that may be randomly organized in space or time, diminishing order overall. We've seen this before in the entropy of water. Water prefers to be random in position, and this is an important driving force in biochemical reactions. And you will recall that delta is a symbol that means change. And so, this can be abbreviated delta S, the universe, is equal to the delta S of the system plus the delta S of the surroundings, and delta S of the universe always increases, so therefore, delta S of the universe must always be a positive number. We have seen this in our own lives countless times. There is a tendency towards entropy. If you do not put energy into a system, for instance, if you don't put energy into keeping a house up, it's going to crumble. And so the laws of thermodynamics are shown here again. The first law is that the energy of the system and its surroundings is constant. Energy can't be made or destroyed. The second law of thermodynamics is the randomness or disorder, the entropy of a system, and its surroundings always increases. The entropy of the universe always increases. You must know these laws. So, how do people figure out that this is actually true? How does one measure the energy of a system? It's really hard to measure accurately heat, pressure, and volume, but you can measure changes. And so, you can measure the change in heat, and pressure, and volume. And because of the interrelatedness of energy and work, you can also measure the work done by the system and the work that's done to the system. And so when people were trying to figure out these laws of thermodynamics, they looked for changes in heat or pressure or volume. One of the first people to look at thermodynamics was a man named Denis Papin. In 1679, he built a tightly sealed vessel that could be used to measure the volume changes of a gas when the gas was heated. All right, so let's take a look at this thing. It's pretty wild. You light a fire in this chamber here, shown by the red arrow. And that heats the water in a closed vessel, and that lifts the lever with a weight. And so the energy of the fire is used to heat the water, and the water is used to raise the weight. Now, unfortunately, when you're dealing with an increase in pressure in a sealed container, well, you know what's going to happen. Yeah, it exploded. This could be prevented by venting off some of the heated gas, a little bit at a time. And the vented gas could even be used for work. And this was the idea behind steam engines. They were soon developed to derive work from energy. And we use the same principle when we catabolize food sources to generate energy. By yielding a little bit of energy at a time, we have a good chance of getting the energy that we need to do work without losing a lot of that energy to heat and to entropy. Let's take a closer look at that. The energy of work is converted in part to heat and in part to entropy. Joule grew up in a family of brewers, and because of the exact precision that goes into making a really good batch of beer, Joule was really good at making measurements. And it's said that while he was on his honeymoon, he said, "If my views be correct, a fall of 817 feet will of course generate 1 degree of heat, and the temperature of the river Niagara will be raised 1/5 of a degree by its fall of 160 feet. Pretty romantic. So he said that some of the energy that's produced by the falling of this water at a long distance was converted to heat. We know it's also converted to entropy, or the randomness of the water molecules at the base of the falls. And so as with the vent in Papin's bone digester, by inserting a water wheel, some of this energy can be harvested to do work, and less will be lost to heat and to entropy. And here's the other person behind the first law of thermodynamics. We saw Joule, and here is Mayer. And Mayer is the person behind the statement that energy is neither created nor destroyed. Mayer also proposed that plants convert light into chemical energy. This chemical energy is stored in the bonds of carbohydrates and other biomolecules made by the plants. And oxidation of these carbon-based fuels is a primary source of energy for life. So let's talk about this for a minute. The energy of photons in sunlight is converted to the chemical energy in chemical bonds by photosynthetic organisms. These are called phototrophs. They're plants and photosynthetic bacteria and archaea. This chemical energy is stored in high-energy molecules such as ATP and NADPH, and then it's used to power the reduction of carbon dioxide from the air. The reaction is shown here, where N is a multiplier. So CO2 plus water yields carbohydrate plus oxygen. Carbon dioxide is therefore a reactant that's taken in from the environment, and oxygen is given off as a waste product to the environment. The energy stored in these carbohydrates is oxidized to supply energy required for life, and also to provide the carbon that's necessary for life. The fixing of carbon from carbon dioxide also provides the carbon that's necessary to build biomolecules. And so, both the energy and the carbon that comes into living systems largely comes in through photosynthesis. Photosynthetic organisms make these things. Herbivores eat the plants, carnivores eat the animals, and omnivores eat both plants and animals. So we alluded to this when we were talking about Papin and his bone digester. Let's go over it again. Glucose, with a chemical formula C6H12O6, is a carbohydrate that can be oxidized to carbon dioxide and water to release energy. The energy that's released, delta G, it's the energy to do work, is 2,870 kilojoules per mole. Holy moly! If burned all at once in a calorimeter, much of this energy is changed to heat and entropy. But if it's released in small steps, however, more energy can be kept in a usable form for work in the cell. And that's what we do. We release a little bit of energy at a time as we break down biomolecules to make energy. This brings up two questions. What do we use this energy for? And what is usable energy? Energy from food is used for heat, to power chemical reactions, and also to do work. Some of this work is mechanical motion. For instance, dynein-powered vesicular transport. This is getting vesicles from one place to another on a highway of microtubules in the cell. It's also necessary for unwinding DNA strands by enzymes called helicases that do this unwinding. Mechanical motion is also motion of muscle contraction that helps us to get from one place to another. A lot of this energy is also used for things that you wouldn't even think about, like the active transport of ions and other solutes across membranes. This is necessary in order to maintain a proper cell environment, like pH, ionic concentration, and the concentration of metabolites in small molecules within the cell and within subcellular compartments within the cell. The processes of replication, transcription, and translation that make DNA, RNA, and protein are processes that all require energy. And so, energy is required for diverse cellular processes. This usable energy is called free energy, delta G, and it can be calculated by the Gibbs free energy equation that's shown here. You're going to have to memorize this equation. It's delta G is equal to delta H, which is a change in enthalpy, minus the temperature, T, times delta S. This equation was developed by Josiah Willard Gibbs, and it's called the Gibbs free energy equation. And the Gibbs free energy equation predicts whether a chemical reaction or a change in physical state will occur spontaneously at a constant pressure and volume. This constant pressure and volume is important. It's an important prerequisite for being able to use this particular equation. Luckily for us, living things are generally at constant pressure and volume, so this works. And a good way to remember this is with the mnemonic that's shown below. Fish is hell without tartar sauce. Delta G is the change in free energy, which is the energy to do work. Delta H is a change in enthalpy, and we said that at constant pressure and volume, you can consider this to be internal heat of a system. T is a change in temperature that's measured in degrees Kelvin. Delta S is a change in entropy, and we know that entropy is a measure of randomness. But how do you use this equation? Well, you plug in values for delta H and delta S, and generally you know the temperature at which the reaction is occurring. And so when you plug these values in, you get a delta G. This delta G can be negative, or it can be positive, or it can be equal to 0, and each of these things means something different. When delta G is less than 0, it's a negative value, and this means that the reaction is energetically favorable and it will proceed spontaneously. Free energy is being released. On the other hand, when delta G is a positive value, when it's greater than 0, this reaction is not favorable as written. It's not going to proceed spontaneously. This means that energy must be put into the system for this reaction to occur. And when delta G is equal to 0, the reaction is at equilibrium, and there's no change in free energy. In general, reactions are more favorable when there is sufficient decrease in enthalpy, when delta H is less than 0, and when there's an increase in entropy. By plugging values in, you can see that sometimes you can get an increase in enthalpy if the increase in entropy is sufficiently high to counteract this. Also, you can get a decrease in entropy if there's sufficient decrease in enthalpy to cover this. But does this mean that if you decrease the entropy, that you're breaking the second law of thermodynamics? No, it doesn't, because this is just looking at the entropy of the system, not of the surroundings. The system and the surroundings taken together must give rise to a positive change in entropy. Not the system alone, and not the surroundings alone, but a combination of the two. The Gibbs free energy equation allows one to determine the energy that's available in a given reaction to do work. The Gibbs free energy equation answers thermodynamics questions like these. Is the given reaction favorable enough to occur spontaneously? If the reaction is spontaneous, how much energy is released during this reaction? If the reaction is not spontaneous, how much energy must be added to the reaction for it to proceed? And finally, what are the relative concentrations of the products and reactants at equilibrium? Thermodynamics does not answer a question like this, however. What is the rate of the reaction? There's no time variable in thermodynamics. Just because something can happen does not mean it's going to happen at a biologically relevant rate, and we'll talk about what this means when we talk about enzymes. Thermodynamics just says, yes, this is a reaction that is possible, given the delta G. For instance, if you were to leave a book outside, it would eventually decompose until you wouldn't be able to find a trace of it. But it's going to take a long time for that to happen, possibly years. So just because something can happen doesn't mean it's going to happen at a rate where you can notice it. So let's do a problem together. This is sample problem number 4 in chapter 1 of your book. Given the following parameters, can this reaction proceed spontaneously? T is equal to 298 Kelvin. Delta H is 84 kilojoules per mole, and delta S is 125 joules per mole Kelvin. And since delta G is equal to delta H minus T times delta S, the question is asking if delta G is a negative number. That means it will proceed spontaneously. So we're going to plug these numbers in. 84 kilojoules per mole minus 298 degrees Kelvin times, now we have to make sure that we do the conversion, times 0.125 kilojoules per mole Kelvin. Did you catch that? We just moved that decimal place over to the left by 3 jumps, 1, 2, 3, to go from joules to kilojoules. We're going to do some math. 84 kilojoules per mole minus 37.25 kilojoules per mole, and we end up with 46.75 kilojoules per mole. This is a positive value. The reaction is not spontaneous. So let's look at this from another angle. Reaction 1 is the reaction going in the forward direction, where the reactants are A and B going to the product C plus D. Reaction 2, on the other hand, is going in the opposite direction. Same reaction, opposite direction. So the reactants are C and D, and the products are A and B. We're going to flip things backwards. So delta H for the first reaction is 84 kilojoules per mole, and the second one it's minus 84 kilojoules per mole. And the same thing goes on for all of these values. The ones that were positive are now negative. So for reaction 1, the reaction is not spontaneous. In fact, it's endergonic. It is going to require energy be put into the system for it to go forward. 46.75 kilojoules per mole, in fact. Reaction 2, on the other hand, is spontaneous, and it's exergonic. It's going to release 46.75 kilojoules per mole in the reaction. When a reaction in the forward direction is not spontaneous, this means the reaction in the reverse direction will be spontaneous. This means that the tendency will be for that reaction to go backward. The reverse reaction is spontaneous. So in going from reactants at a higher energy and or a lower entropy to products of a lower energy and or higher entropy, the force is with you. This reaction will occur without an input of energy. For the reverse to occur, however, an input of energy must occur. Negative delta G really does rule the world. So before we go any further, there are some terms that we're going to have to define. These are delta G naught and delta G naught prime. Now, delta G naught is the free energy of a reaction that's carried out under standard state conditions. This is at 25 degrees Celsius and at 1 atmosphere, starting with 1 mole of each reactant, and then letting the reaction proceed as it will. Delta G naught prime, however, is something different. It indicates that the reaction occurs under standard conditions, but it's more similar to what happens in biological systems. We're still at 1 atmosphere, still at 25 degrees Celsius, but we're at a pH of 7, an aqueous solution. Here the molar concentration of water is 55.5 molar, and we're going to assume ideal conditions in which the charge of water does not affect the reaction. So, we know what delta G naught prime is in a beaker, and that's very well and good. We can do the reactions in the laboratory in aqueous solution, 1 atmosphere, pH 7, 25 degrees Celsius, wonderful. But things are different in a cell, such as this epithelium that's found in the respiratory tract. In living things, the conditions are different. In living cells, the relative concentrations of products and reactants change. They change in response to environment, both internal and external. And so you can start with what you know about standard state conditions, the reaction happening in a beaker, but you have to account for the relative molar concentrations of the products and reactants when you start the reaction. And so for this reason, the things that occur in a cell are very different depending on the internal conditions of the cell itself. And so what you really want to know is not what delta G naught prime is. That's what happens in the beaker. What you want to know is what is delta G? What is the free energy that's given off, given that you're going to have different molar concentrations of reactants and products when you begin this reaction. Let's take a look at this a little more closely. So for a given reaction that occurs in a cell, A plus B going to C plus D, we have to take into consideration several things to be able to find out what delta G is. And so we have delta G is equal to delta G naught prime, and that's similar to what would happen in a beaker. These are under standard conditions. And here comes a little bit of adjustment that we do for cellular processes. Plus R times T times the natural log of the molar concentration of one product times the molar concentration of the other product divided by the molar concentration of one reactant times the molar concentration of the other reactant. Okay, so what's R and T? R is the gas constant, which is equal to 8.3145 joules per Kelvin per mole, and T is the temperature in Kelvin. This is important to consider because some biological reactions reverse direction as a result of the concentrations of products and reactants that are found in a cell. So you can have the delta G naught prime in the beaker be one thing. You can even say that that reaction could not go under these conditions. However, in the cell, you can actually force that reaction to go. And that's what we do, really. We'll see evidence of this throughout the semester. Now, the natural log ln can be converted to log base 10 by multiplying by 2.303. And so we come up with the equation delta G is equal to delta G naught prime plus 2.303 RT natural log of the molar concentrations of the products over reactants. So let's look at the isomerization of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. The delta G naught prime for this reaction at equilibrium is plus 7.53 kilojoules per mole, and so in the beaker you will not have dihydroxyacetone phosphate yielding glyceraldehyde 3-phosphate. Instead, you'll have glyceraldehyde 3-phosphate yielding dihydroxyacetone phosphate. This reaction is going to want to go in the reverse direction. But this reaction happens all the time when we break down glucose for energy. How does this happen? Well, if we look at the equation below and plug in the molar concentrations of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, as they are generally found in the cell, we come up with 7.53 kilojoules per mole plus 2.303 times the gas constant times the temperature times the log of 3 times 10 to the minus 6 divided by 2 times 10 to the minus 4. And when we do the math, we come up with delta G as being negative 2.89 kilojoules per mole. And so this reaction certainly will go in the forward direction. And this is how biochemistry happens in our bodies. We can modulate the molar concentrations of products and reactants to make reactions go in the direction we want them to. And this is an important thing to remember. Hi. Today we're going to talk a lot about math. Thermodynamics. There's a lot of math in thermodynamics. Are you afraid of math? Please don't be afraid of the math. Fox says don't be afraid. You don't have to know the conversion factors. You don't have to know the constants. But what you do have to memorize is the Gibbs free energy equation and the Henderson-Hasselbalch equation from the previous lecture. Listen to the lecture, do the practice problems, and if you have any questions, just drop me an email.